Okay, so welcome everybody to today's seminar of the series called Cutting Edge Advances Towards Personalized Mental Health, hosted by our Center for Precision Mental Health and Wellness. I am Dr. Leonardo Tozzi, and I am a co-director of this seminar series, along with Dr. Xue Zhang. I'll just start with a couple of housekeeping items. The first thing is the format for this presentation, which will be a few minutes for introductions, then followed by a talk by Dr. Penninx, which will be around 40 minutes, and then we'll leave 10-15 minutes for Q&A. And about the Q&A, I would like to ask all participants who have a question to put it in the Q&A option in Zoom. And at the end of the talk, I'll just go over the Q&A section and ask our speaker to answer the questions that are there. So with that, I would like to hand over to our director of our center to introduce our special keynote speaker. Thank you very much.
Thank you so much. So welcome everyone. I am Leanne Williams, Director for the Center for Precision Mental Health and Wellness here at Stanford University. And I'm delighted to welcome you to this seminar series on cutting edge advances towards personalized mental health. My center launched this seminar series to hear from world leaders whose findings are creating the new field of precision mental health. And our wonderful co-directors, Dr. Tozzi and Zhang, have organized some truly exceptional speakers. For those of you new to our center and the seminar series, I invite you to learn more and to join as a member, and the web links are here on this slide. And today is a truly great privilege to introduce Professor Brenda Penning. Professor Penninx is a global leader in changing the way we understand and treat depression and anxiety. Her outcomes are making a difference at the population level and for individuals. Professor Penninx is based at the Amsterdam Universitair Medische Centre, connected to the Vrije Universiteit. She is the principal investigator of a landmark study, the Netherlands study of depression and anxiety. And if you look it up, you'll find it as NESDA at nesda.nl. And Professor Penninx has led NESDA since 2004. The scientific focus is on the etiology, cause, and consequences of depressive and anxiety disorders. She translates observational research findings in novel intervention studies. She supervises a research group of more than 25 PhD students, postdocs, and assistant professors. And Professor Penninx has an outstanding record of excellence. She's on the list of top women scientists, ranked 6th in the world and 1st in the Netherlands. She has published over 900 scientific papers, and her impact is recognized by her membership of the Royal Dutch Academy of Sciences, of which she serves as vice president since 2022. I've been a long-standing admirer of Professor Penninx's work. I'm glad to have had the recent opportunity to unite data sets across our centers, and I thank her for this wonderful opportunity. And today I welcome Professor Penninx, who will present on immunometabolic depression, from conceptualization towards implementation. So welcome, and we look forward to your presentation.
Thank you very much, Liana. Let me share my screen. I hope everybody can see my screen. And it's really a wonderful pleasure. It's late in the evening, but this, I think, is really one of the advantages that the COVID pandemic has created, that webinars are now much more common and that we can interact even at these times across the world. So it's a pleasure to talk to you about immunometabolic depression, from conceptualization to implementation. But before I do so, it's good for me as an epidemiologist to really focus the attention on why research in the field of psychiatry, and especially the focus of depression, is so important. And I think this is clearly illustrated by the worldwide disease burden as the WHO estimates them every few years. This is an index of the years lived with disability and the impact that specific disease groups have. And this is maybe a little bit a slightly older picture, already coming from more than 10 years ago, but the shape of this pattern has not changed much. And as you can look at this picture, you see both for men and women, a big, large red area, which is the impact of psychiatric conditions on a very important disease burden outcome, the years lived with disability. And it is really psychiatric disorders, mental disorders, that have already quite early in life a massive impact on disease burden. That's why I think it's so important to do proper research in the field of mental health. As you see over the lifespan, the impact of psychiatric conditions becomes a little bit less in terms of relative comparison to other disease groups. And that is really because lots of the other diseases really arrive at late age. And we all know that mental health disorders are not, they are arriving very often at an early age already. Also, if you look at another outcome, disability adjusted life years, the impact of depression is huge, and it's ranked in the top 10 of disorders with high impact on disability adjusted life years outcome. But not only that, I think this is a picture coming from a Lancet Commission paper we published last year, which shows that actually the pattern that the impact of depression has on this outcome has been really stable for the last 30 years. And as a comparison, if you look at the impact of ischemic heart disease, it has been possible to decrease the impact of ischemic heart disease on disease burden in the last 30 years. But this blue line is really stable over time. And I think it's really an important picture to capture, because what does this mean? This really means that despite the fact that we have treatments, that we have good research going on, it has been really a challenge to impact on the disease burden of depression. So what are the reasons why the impact on public health is so large, that depression has such a big role here? Well, it are, I think, a few essential characteristics that we know depression has. As I mentioned already, it's impacting already early in life and can therefore have chronic impact during a really long time period. It strongly impacts on the functioning of individuals. It is quite common with a current prevalence of around 5 to 6% and a lifetime prevalence that is at least double. And it has very often a chronic picture for lots of patients. Also, we know depression is interacting strongly with somatic health. So it's just not only the impact that depression has on mental health and suffering, it's also the negative impact it has on somatic health and somatic disease outcomes. However, considering all these factors and considering that the disease impact of depression is stable in the last 30 years, we need to identify underlying problems. And as you may be well aware of, we know there is still suboptimal treatment. Both there are people not recognized and not being treated at all. But we also know that even if people get treatment, they are not always fully recovering. And there is lots of trial and error going on in clinical practice. We know the one-size-fits-all approach is not correct, but it's difficult to get to the personalized treatment options that work better in depression. And I'm sure that in your center, you're doing lots of research in this field as well. So I think this is probably an introduction that most of you are very much aware of, and that we are addressing too in our research line in Amsterdam. So think about depression and think about the concepts that we consider when we diagnose depression. These are the central DSM criteria to indicate depression. And lots of these symptoms are quite diverse, not in the fact that they come from different dimensions. So some of these symptoms are tapping emotions, others are tapping cognition, and others are tapping somatic symptoms. But not only that, it's also quite amazing that some of these symptoms actually can go in both ways. They can go to an increase or to a decrease of an underlying dimension. So think about sleep. We do see depressed patients that have hypersomnia, and we have depressed patients that have insomnia. And there are other examples. Think about appetite and weight. They can go up or they can go down. And same is true for psychomotor retardation versus agitation. So to me, it doesn't make any sense that there is one underlying pathway that we can expect to yield this diverse type of symptom outcomes. And it has been calculated that if you really add all the combination possible of symptoms that arrive at the diagnosis of depression, then there are more than 200 combinations of symptoms possible. So this already indicates the clinical heterogeneity of depression. However, we do know quite a lot also, despite the fact that this concept is so heterogeneous. And I think we really should use this as a starting point for research. Well, first of all, we know there is an heritability of depression. And as you compare depression to other psychiatric conditions, the heritability of depression is quite low. It's really on the lower end of all the psychiatric conditions, but still about 35% heritability has been estimated in multiple twin studies, for instance. So we know there is a genetic component. To really get better to the basis of where this genetic component comes from, we do genome-wide association studies. And this is a picture from one of the latest studies where they included more than 240,000 cases and over 500,000 controls. And this is what you see here is the Manhattan plot showing the significant various genetic loci that are significantly related to the risk of depression, showing that depression really has a polygenic genetic basis. And I think from looking at some of these top genetic findings, it's clear that the genetic basis of depression, first of all, is connected to neurological and brain function, but also to the immune system and to obesity and metabolomic related pathways. That's where some of the top hits lead us to. So also this genetic study, as I mentioned, indicated that genetic variants identified as being relevant for depression are heavily involving brain pathways. And I know your research group is doing quite a lot of work in the field of connecting brain mechanisms to depression. And we know that there are differences between depressed patients and controls, both if you look at structural, but especially when you look at connectivity and functional activation patterns that are really indicating that depression is indeed involved brain dysregulations, especially at certain brain circuits. But it's not only that. I think really depression should be not just considered a brain condition, but should be considered a whole body disease. And I think that is really clear from looking at the picture of the biological stress systems that our body has, for which the brain is heavily important, but the brain is connected to the rest of the body for the functioning of all these three biological stress systems. And if I think about the biological stress systems of our body, then I think of three systems. The HPA axis that lots of people of you probably are aware of, the axis that creates the stress hormone cortisol. But also the autonomic nervous system, which is the most direct activated stress system our body has and responds, for instance, with increased heart rate and decreased heart rate variability. So it puts the body in a fight and flight mode when people are exposed to a stressor. But also our immune system is a central biological stress system that our body has. It's not the most immediate. It takes more time than the other two stress systems, but it's also being activated when the body is exposed to stressors, and that can be physical stressors such as viruses or bacteria, but it can also be emotional chronic stressors. So these are the three stress systems that directly are linked to brain activation, but are also connected heavily to cardio and vascular and metabolic systems. So it's really clear from this picture that depression is not just only a brain disease, but really a whole body disease. And we know that this is working, visible in lots of the metabolic conditions that can be examined. And there are actually multiple cross-sectional as well as longitudinal meta-analysis indicating a link between, for instance, depression and obesity. And there are multiple indications showing that depression is linked to an increased risk of leptin and insulin resistance, for instance. So this is not just only impacting on stress systems and metabolic systems, but this is really driving our biological aging clock. And as we indicated in some studies conducted in our NESTA project, we looked at cellular age indicated by telomere length, for instance, and we looked at epigenetic age, which you can get from DNA methylation. And we just simply, by calculating these aging clocks, you can compare the biological age of people with depression and without depression. And in both these examples, we did confirm that the impact of these immunometabolic dysregulations and stress system dysregulations that can be seen in people with depression, that they are potentially really responsible for a more general underlying, more advanced biological aging, because we confirmed that people with depression had significantly shorter telomeres than healthy controls, and that they had more advanced epigenetic aging clock as well. Well, if you summarize some of these biological, pathophysiological indicators that I just summarized, I think it's, first of all, very clearly visible in this table that this evidence is not just coming from single studies. This is coming from many, many studies across the world that are confirming that these pathophysiological mechanisms are linked to depression, as you can see from the column indicating the number of studies that have looked at these pathophysiological mechanisms. But the second observation from this table is that despite the fact that overall there is quite consistent evidence for associations, pooled effect sizes for none of these pathophysiological mechanisms are huge. So for instance, even things like the HPA axis dysregulation, which I think has been in textbooks for already 20 to 30 years, if you really look at meta-analytic evidence, the Cohen's D's of associations between, for instance, the stress hormone cortisol and depression is never exceeding an effect size of 0.3. So that really means that we are really talking about modest associations here. And that's also reflected in the third column that is important to emphasize here, and that's the heterogeneity column. These are the heterogeneity findings coming out of all these meta-analyses that have looked at these pathophysiological mechanisms in depression. And as you can see, heterogeneities across studies are huge. Well, that could partly be reflected by the fact that studies have used different methods, have used different samples. But I would argue that it's also the heterogeneity of depression itself that is adding to this heterogeneity of associations. And that's really why we in the Netherlands study of depression and anxiety have set up a large cohort, including lots of depressed patients, so that we can really go a little bit more into depth on what is the heterogeneity within this patient population. So the design of the NESTA study is actually a naturalistic cohort study where we started out with over 3,000 individuals. And we're still following these people over time. We are almost finished with our 13 follow-up wave. So this means we are following these people already for 13 years of time, so that we really have lots of information on their development and their chronicity of mental health, but also their somatic health development, for instance. Participants in our project, they came from diverse settings because we really wanted to reflect the entire range of the population as it occurs. So they came from community, general practice settings, and mental health care settings. And they were at the time when we collected our data at baseline, they were in their 18 through 65 years of age. As you can expect, we have a female preponderance in depression. So you have a larger group of women than men when you conduct these kind of studies. This is really a national study. We collaborated with 3 university medical centers in the Netherlands and lots of mental health care organizations. And so far in the last 20 years, there have been lots of publications based on this project and lots of PhD theses that have been finished using data from the Netherlands study. So we really have created what I call a research infrastructure to do depression research. Oh. And one of the first things we did, and I'm going to show you some of the findings that we started out with when we were interested in this heterogeneity of depression. So I think one question you can address is, and it's not a very easy question to address, is where do you start when you want to study depression heterogeneity? You can start from the data, and you can start from your theory. You can start from the symptoms, and you can start from the biology. Well, I'm just going to give you an example where we started with the symptoms, but actually we also have conducted lots of analysis since then where we started with the biology. And in fact, we really arrive at the same picture, and that's the picture that I'm going to show you now. So this is the first, one of the first analyses we did within over 800 depressed patients. So we really only took people who were in a current depression. Some of these people were using antidepressants, others were getting psychotherapy, and some of these people were actually not treated. So it's really a naturalistic cohort. And what we did address, or what we asked ourselves in the beginning was, if you just look at the symptom prevalence that these people have, do you see certain clusters of patients where patients have more similar symptoms than in the other clusters? So you can do these data-driven approaches where you just look at the clustering of symptoms to see what are common symptoms that you are often co-occurring. And we did analysis and found that there were actually 3 symptom clusters that we could identify. So the first symptom cluster is a little bit a more moderate or more milder depression class, which is the green line, which actually were identifying people who had just slightly lower scores on lots of the symptoms that we looked at. And then there were 2 other classes that I call here severe typical and severe atypical. They were both quite common. They did not differ in the overall depression severity. They did not differ in the overall disability and in the overall duration of symptoms. They just differed in the types of symptoms they had. And to put these 3 classes in a more readable table, this is what I then come up with. So we have a more moderate class, which just has less severe symptoms. But then there are 2 severe classes that really, if you look at their overall inventory of depressive symptomatology score, would qualify as at least moderate to severely depressed. And these people had slightly different types of symptoms. So the severe typical people were most scoring highest on depressed mood, on weight loss, on decreased appetite. They were poor sleepers, so they had less sleep. They were most suicidal, had poorest quality of mood, and had most often early awakening. And some of these symptoms you may recognize as what the DSM calls melancholic depression. However, DSM also qualifies some other symptoms as indicators of melancholic depression, such as psychomotor disturbances and diurnal variation, but these were not more common in our cluster. So these were not by our data-driven approach, indicators that were different between the severe typical and the severe atypical group. Then we have another cluster, which was common in about 1/4 of all the depressed persons, that scored a little bit more on the atypical symptom spectrum. They had weight gain, they had increased appetite, they had more sleep, and they were scoring very high on fatigue and energy loss and on lead and paralysis. And again, these are some central symptoms that the DSM classifies as atypical symptoms. But again, interpersonal rejection sensitivity and mood reactivity, which are also listed in the DSM, were actually not indicators that were listed or that came up as being relevant in our data-driven approach. Well, what we did subsequently is we went back to data and classified these two severe depressed groups a little bit better by just looking at what are other correlates that we can find in these two different groups. So we know they are different on certain of their symptomatology, as I listed here, but we also did find some other differences. The typical depressed people were a little bit more often smokers and had more negative life events and more childhood trauma. The people with atypical depression, on the other hand, had a slightly earlier onset and were slightly more often female. So these were not huge differences, but some subtle differences in characterization. And then again, typical depressed people, if you look at their course and at their clinical symptomatology over time, they showed more persistent comorbid anxiety and higher suicidality. And it were the atypical depressed people that really had a poor course when you look at their metabolic syndrome profile and when you look at some of their somatic outcomes. So you can kind of wonder, you know, is this kind of a different pathophysiology? That's what we started to look out for a little bit more, because in our project, we actually have measured quite some of the stress system functioning that I just mentioned in the beginning. And we have measured, for instance, immune markers such as CRP, IL-6 and TNF-alpha, but also other indicators of metabolic syndrome functioning. And on the upper part of this graph, the blue dots, you do see that there are some subtle, slight differences between people with depression as compared to healthy controls, but not for all these markers. In fact, the people with depression were having a slightly higher BMI and a slightly higher waist circumference, but on the other aspects, there were no significant overall differences. However, if you would repeat this comparison only for the atypical depression group, then you see much larger differences as compared to healthy controls. And that's shown with the red dots, where you now do see that on actually all of these immunometabolic markers, people with atypical depression scored worse as compared to healthy controls. So this is indicating that the immunometabolic dysregulation that we have measured in our project were mainly seen in people with atypical depression. And if you compare people with typical depression, so the people who had a little bit more of some of those melancholic type symptoms, we actually did not find any differences with people who were healthy controls. If we just go further and use the blood samples to really measure in more detail some proteomic signatures that we can measure in detailed analysis these days, where we measured more than 170 serum proteins that cover differential hormonal, immunological, and metabolic pathways, then we can again see discrimination between people with atypical versus typical depression. And this is directly comparing those two groups of depressed people, where we do see, for instance, that atypical depressed people are the ones who score higher on insulin, higher on CRP, and higher on leptin, which was really in line, for instance, also with another finding we have when we look at leptin resistance, which turned out to be much higher in people with atypical depression. So just some indication that also we really, when looking at biological markers, it is possible to discriminate between these two types of depressed people. And another picture that shows kind of similar patterns is when we measured lipidomics, when we measured metabolomics, but really mainly covering the lipids dimension, using the Nightingale Health platform that measures 51 markers that are really basically measuring lipid function. Again, you see on this figure that if you look at the sum score of overall depression severity, which is the blue dots, there are only 2 significant associations between metabolomic markers and the severity of depression. However, if you only would calculate the atypical symptoms that are very relevant in atypical depression and look at the sum score of those symptoms, you do see much stronger associations with now 31 significant findings with links to metabolomic functioning. So indicating again that lipidomic measures are much more strongly related to atypical depression than with overall depression severity. So the types of symptoms that people have really matters. This is a slide showing that it is not just us finding some of these associations with immunometabolic markers. So I put several references here to other types of studies and also to other studies who started from different approaches, where they didn't start with symptom class analysis, where they, for instance, looked at what are the symptoms that are linked to certain biological dysregulation. So they started with the biology. And I think there is really kind of a more and more emerging consensus in the literature that it really matters what types of depressive symptoms are apparent, at least for dysregulations in immunometabolic profiling. So I think we can put the box a little bit further here and fill in that also at the pathophysiological level, there are actually some different underlying pathophysiological mechanisms that distinguish these 2 groups of depressed patients. And what I didn't show you here, but we have published in Molecular Psychiatry, a paper by Femke Lamers, is that people with typical depression are the ones who actually showed the highest hyperactivity of the HPA axis. We have measured saliva cortisol awakening responses in almost all of our patients. And we do confirm that hyperactivity of the HPA axis is more common in depression. But if you compare typical and immunometabolic depression, which is how we now call atypical depression more and more, it is really the typical depressed people that show more evidence of hyperactivity of the HPA axis than it are the people with immunometabolic depression. So then we went actually further because we also wanted to know, can we get more evidence from a genetics perspective to really further confirm and also further test the causality of our findings? Well, first we looked at gene expression because we have measured gene expression in almost all of our participants. And with gene expression, you can actually identify of inflammatory pathways. And we confirmed that, again, if you look at enriched inflammatory pathways, they were much more often prevalent in people with atypical depression than they were in people with typical depression. But also GWAS, the genome-wide analysis and the genome-wide genotyping that these days is possible, is a really nice tool to further test underlying associations. So you can actually take results from lots of genome-wide studies, even outside of the field of mental health, and take summary findings of those GWAS analysis to indicate a polygenomic risk score, which is basically identifying the genetic vulnerability for a specific trait. And that's what we did in our project too. And for instance, took and created polygenomic risk scores for schizophrenia to get information on a completely different psychiatric condition, but also create polygenomic risk scores for body mass index, for instance, and for inflammatory markers. And when we did do that, it was again clear that the genetic underlying vulnerability for typical and atypical depression is different as well. It were people with typical depression that scored the highest on the schizophrenia polygenomic risk score, so they share more with the genetic vulnerability for schizophrenia, Whereas if you look at people with atypical depression, they scored really high on, for instance, a polygenomic risk score for body mass index. And we extended this even using the Psychiatric Genomics Consortium data or the UK Biobank data to further confirm these findings for instance, the genomic risk score for CRP, but also for leptin. And again, the picture is quite stable, and emerging here is that the genetic vulnerability of atypical and typical depression follows the same pattern as the underlying pathophysiology that I just indicated. Well, maybe this is, I think, where we are right now. That's not only these two types differ in symptomatology, they differ in correlates, they differ in cores, they differ in the pathophysiology that is underlying, and they differ in the genetic basis. So this is a picture that I think really comes out of our research, where we really look at the different layers of really going from genes to biology to clinical correlates that we can put together in this picture. And we believe that especially for this immunometabolic depression, there is more and more evidence that there is a clustering of specific, what we call atypical, and they're really kind of energy-related symptoms that are very prominent in their link with underlying immunometabolic dysregulations. And that can really also obviously tie a condition like major depressive disorder together with metabolic disorders, such as cardiovascular disease, diabetes, and obesity. And we know that these conditions are strongly linked, and that the link between those conditions is really bidirectional, that depression is increasing the risk of developing these metabolic diseases, and metabolic diseases are increasing the risk of developing depression. And the underlying shared pathophysiology can really provide a very clear explanation for this. Well, I want to use the last 5 minutes to address the question, why is this all relevant? Why are we doing this? Well, I think if you really want to bring it to the clinic, you also should obviously show that this could potentially be important for the clinic. Well, first of all, there is evidence from a meta-analysis that has been published 2 years ago that shows that it could well be that immunometabolic dysregulation, such as inflammation and metabolic syndrome, is actually in the way of antidepressant response. And there are some findings that indicate that people who have these immunometabolic dysregulations are having poorer response to antidepressant treatment. And then obviously another reason to be enthusiastic about these findings is that immunometabolic dysregulations can be targeted in different ways. We do actually have medications out there that target and reduce immunometabolic dysregulation. So really the question is, can we use them better for this specific group of immunometabolic depression? And could that yield a potential differential and maybe kind of add-on treatment option that we haven't explored for our patient group? And another thing is, obviously, if you think about immunometabolic dysregulations, we actually do know that lifestyle interventions, several out there, have been tested and have been shown to reduce immunometabolic dysregulation. So just a few slides on what we know from literature on, for instance, the use of anti-inflammatory treatment for depression. And there are actually a few meta-analyses out there that have looked at the question, if you treat people with anti-inflammatory treatments, is that good for depression? Is that reducing depressive symptomatology? And these are two meta-analyses that have summarized data where they looked at different types of anti-inflammatory treatments. They have compared them usually to placebo. And the overall conclusion is that for several of these anti-inflammatory treatments, there is some suggestion that they are beneficial in the sense that they are reducing depressive symptomatology. Well, this looks quite promising, but I have to say that almost none of these studies have been conducted in psychiatric settings. This is mainly evidence coming from rheumatoid arthritis patients, where they are really treating people commonly with anti-inflammatory treatments, for instance. So it has not been tested in psychiatric practices in large settings. So that is really some first clue or some first glimpse at maybe some promising evidence, but we really need to bring it to the clinical practice if we want to be certain that this is going to be effective for our patients. A second thing is that none of these studies that I have showed you and that have been conducted, because there are now a few studies that have been taken this evidence to the clinical practice and tested whether depressed patients can benefit also from anti-inflammatory treatments. But almost all the studies that have been conducted so far, and they are really small scaled, but they have never selected people with depression and certain features. They have not selected those people who actually started out with inflammation. And from just what I've shown you, we know that that is probably not more than 1/4 of all the depressed people. Not everybody has immunometabolic depression. So this is one of those studies that have been conducted in the UK in a group of only 39 patients, where they treated people randomly with either minocycline, an anti-inflammatory agent, or a placebo. And the overall conclusion of the project was that there was no difference. However, when they started doing stratified analysis and looked then at people who did and who did not start with inflammation by just looking at a very crude indicator of CRP, then they only found that the effect was much larger in people who started out with high CRP levels. So maybe only in people with inflammation, we could even expect to find an association and a beneficial impact on depression by treating people with anti-inflammatory agents. But again, this is a very small scale study, and we need better proof in order to implement such a finding in real life. So that is one a trial that we are currently working on as well. We are currently conducting what we call the inflamed RCT, where we actually have where we are randomizing people to celecoxib, an anti-inflammatory NSAID, versus placebo in a 13-week randomized trial where we measure depression over time. But now we are only recruiting people that will fulfill the definition of immunometabolic depression as how we are identifying it now. So we're not just taking all people with depression, but we really take the people who qualify for major depressive disorder, who have a high IDS score, but also a high IDS atypical energy-related symptom score, and a CRP above 1. So we're really stratifying already at the basis of this trial and are only treating people with anti-inflammatory agents when they are fulfilling our definition, our working definition, I have to say, of immunometabolic depression. Quickly about 2 lifestyle interventions that I really think are very important to consider for the field as well. One is running or exercise. Running, we know, is reducing inflammation. We know that from large trials outside of the field of psychiatry. And we know that physical activity is reducing metabolic syndrome abnormalities as well. So there has not been that much work in the field of exercise in psychiatry, what exercise does for depression, and especially not what exercise does for the pathophysiology mechanisms underlying depression. We can expect that running is beneficial for mental health, but will it be working for all depressed people, or will it be mainly working again through immunometabolic dysregulation restoration? There is one study conducted by the Texas team where they found that running is effective in reducing depression, most strongly in those with inflammation and most strongly in those with atypical symptoms, But again, that's only one study and really small scale. So we need better proof here as well. And then finally, the field of nutrition is huge. It's a difficult field. We have done some nutritional interventions ourselves. They're not always very successful so far, but I do think that this is a very important field as well, because we know, especially through all the work going on these days with the microbiome and the gut, that there is a very strong connection between the microbiome and immune metabolic functioning, which is, really strongly linked to nutritional intake. So I think this is really an area for lots of opportunities to test what potential nutritional interventions and nutritional strategies could do, and to what extent they can be, in addition, beneficial for depressed people above and beyond our standard treatment opportunities that we can give to people. So I come at the end of my talk. I hope that I have shown you just one example, and I'm not saying that immunometabolic depression is the only example. I think there are some other examples out there, but that really helps us to untie depression's heterogeneity. And if you would ask me, what do we need to do as a field to untie this depression heterogeneity? Then I really think we can and should apply deep phenotyping approaches like we are doing. We should use the tools that genetics, omics, and environmental pathways and imaging provides us to better understand what is going on at an individual patient level. We need to move beyond single markers because I think there will not be one single marker that will identify a very specific pathways and identify those who are most treatment responsive. And we can only do that when we collaborate with others, validate, replicate, standardize assessments so that we can really collaborate to untie this quite complex heterogeneity of depression. And with that, I would like to thank you for your attention. And I clearly want to thank the big group of people helping me with Nesta. This is definitely not an infrastructure that I can do on my own. I've had lots of support from lots of good people in the Netherlands and beyond. So it's really a pleasure to show some of that result from our project to you. Thank you.
Thank you so much for a wonderful, wonderful presentation. Leonardo, I think you'll host the questions.
Yes, we thank you so much, Dr. Penninx, for the great presentation. And we do have some questions that came in already. So we have two in the Q&A box. So the first question is, very much related to the talk. Is it possible that depression is a symptom similar to the other symptoms of two or more distinct, though currently not well understood, disease etiologies? So if you probably could say more about the etiologies and what you think about that.
Yeah, so I do think that depression, as I see it, is really a syndrome where there are some clusterings of pathophysiologies and clustering of symptoms, and you can obviously arrive at certain symptoms through very different pathways. And I think there are obviously also some shared symptoms between clusters. So I have been highlighting some of the differential symptoms, but it's probably also good to emphasize that there are lots of symptoms that actually were not differential between these clusters, and they could be completely seen as indeed shared symptoms and symptom dimensions that were not able to differentiate in these two types of groups.
Thank you. And then we have another question by another attendee who says, thanks for a great talk. I was wondering if you were able to disentangle HPA hyperactivity versus chronic activation, such as by using surveys or hair cortisol measurements. I know
Yeah.
HPA acute hyperactivity, sorry, there's two parts. I know HPA acute hyperactivity can be one result of chronic stress, but it can also look like HPA habituation. And it would be interesting to see how chronic stress intersects with metabolic depression.
Yeah, so we have measured hair cortisol and we have linked that also to depression. We do find an association with higher hair cortisol and depression as well. The hair cortisol measure was not assessed at the same wave as the saliva cortisol measures that we had. So I can't really compare the findings that we have in hair cortisol directly with the saliva cortisol findings. We know hair cortisol is a much more chronic indicator of hyperactivity of the HPA axis. But in our case, we have not been able to find very different clinical features linked to hair cortisol. We have published that we could not, for instance, indicate that childhood trauma, which is a clinical characteristic that really probably identifies very chronic depression and already an hyperactivation of the stress system early in life. We could not identify a link with hair cortisol and childhood trauma. So yeah, we have confirmation that also chronic cortisol indicators are linked to depression, but we cannot directly compare it to what we find with saliva cortisol. And for those interested, I could send you some of the papers from the Nesta project where we have confirmed these findings. So I'm happy to take an email from you if you want more details.
Thank you so much. We also have questions coming in through the chat, so I'm going to read them out. So jumping to the chat, I'll try to go in order if I can. This question is by Dr. Pines from our lab. Thank you for speaking, Dr. Penninx. I really admire how your work fills a critical gap by considering the brain in the broader context of the bodily systems it operates within. I'm curious what you think about the brain in the broader context of the social cultural context it operates in. For example, Netherlands residents seem to be particularly susceptible to climate distress, with 4 in 10 residents of the Netherlands reporting being extremely concerned about climate change in 2022. I was wondering what you think are some of the more promising emergent research directions in bridging chronic stress, immunometabolism, societal events, and mental health.
Wow, that's a huge question. And it really brings me, well, the answer to this question really brings me to a new project that actually we started this year, which is called Stress in Action. And actually we got funding to look a little bit more at daily life stressors. So I think what we do quite a lot in our research is we go, think retrospectively on what has been impacting in a person's life. How was a childhood? How was the upbringing and parental bonding and so on? But we're not having very many good measures of what is really stressing people in a daily life context. And what this new project is going to do, and we will use partly the Nesta infrastructure for this. So this is actually for us a new wave in the coming years, where we are really going to measure much more in a daily life context by using ambulatory assessments. So we will basically use phone, use actigraphy, use environmental exposures to really find out on a daily basis what is stressing people and what are people wondering about and how does that activate their biological stress systems and their behavior. I think this will be, it's not a direct answer to the question on climate stress and whether it's true that that is a very critical stressor in Dutch citizens much more than in American citizens. I'm honestly not sure, and I'm not aware of any research that has compared this. So if you have it, I would be really interested in seeing it. And I think as far as I know, America, I think climate stress is probably very heterogeneous within America already. So that is the same in the Netherlands, actually. There are lots of people that are stressing about these kind of things, but they're also stressing about the war. They're stressing about, you know, the earthquakes in Turkey. And so to what extent these stressors that change all the time are really building up on how people are dealing with stress in a biological sense, in a behavior sense, and in a cognitive emotional sense, I don't think we know that very well. So I think that's really something that is very important for our research to dive into a bit more. And whether, to what extent this maintains disorders and symptoms over time, I think is also a question, we need to address.
Thank you so much. I will now combine kind of a few questions that are all in the same, around the same topic, which is basically whether you believe that your current diagnostic criteria for immunometabolic depression can be translated to DSM and how they relate to existing DSM diagnoses. Do they match? Is it possible to expand the DSM, or do we need a shift towards a new quantitative diagnostic framework?
Yeah, I think it is too soon for me to change DSM. I think DSM, you know, and it's only a guideline, but it's really a helpful guideline. And I think we only need to replace it when we certainly know that making different diagnoses matters for the clinical treatments and therefore the course of individual patients. And as long as we don't know that, for me, it doesn't make any sense to change a DSM. I do think that the immunometabolic depression fits quite closely to some of the core features of atypical depression. I also do think that atypical depression has a lot to do with anhedonia. So there are some research groups that zoom in on anhedonia, use different instruments for that. And I think there is really a lot of overlap with the concept of anhedonia. So I think that should be considered a little bit more strongly within this concept too. But I think we only should change the DSM when we can prove that identifying this subgroup really is useful for personalized medicine treatment decisions. And otherwise, we just should continue the research till we can change that treatment strategy.
Wonderful. And we also have a question by Dr. Jang in the chat. Saying, Dr. Penninx, thank you so much for this wonderful talk. I'm wondering if in the longitudinal cohort you have collected neuroimaging data and how you think the immunometabolic subtype might differ from healthy subjects or typical depression in their brain circuits. Maybe the somatomotor network question.
Yeah, so we have longitudinal neuroimaging data. We do not have this on everybody. So our numbers at baseline, we started with 300 and we're down to under the 300 right now. So it's not massively huge, not like in the UK Biobank and not in some of your studies probably. We have looked a little bit at, the numbers, especially when we divide atypical versus typical depression, because I think that's what the question alludes to, our numbers are not huge. So I think this is really something that should be examined better. And I think there are some other groups that do find, for instance, that especially the reward system activity is highly relevant for immunometabolic depression. I think this is, for instance, work by the Felmer group in Atlanta, indicating, especially for people with anhedonia and the atypical depression symptoms, that they do see very strong associations with reward activation in the brain. And there has been some work done looking at resting state functioning and functional activation, but there they have used food cues, which is not being done that much. But this is a study done by the, I forgot the first name. He now works with Janssen. well, maybe later, but and
Brenda,
yeah, well, he now works with Wayne. It wasn't Wayne itself,
Okay.
but a person working well, I come with the name later, but they have looked at these two depression concepts too, and do find very clear differences also in the reward system, but really mainly looking at functional activation, where they activate people by activating their appetite. So that's a very different way of looking at functional activation than we standardly do, I think, in brain imaging studies, where we usually, map on emotion activation. But I think the entire concept of appetite and emotional eating is actually very relevant here as well. And I think especially if you think about depressed people who are overeating, they may have an energy balance problem, but they may also actually be really using their eating partly for emotional comforting. And that I think is probably slightly going on in the immunometabolic depressed subgroup too. And there are some studies we have done a little bit in that field also that actually have looked at emotional eating questionnaires and really see that food intake and the snacking and the comfort food is actually one way of self-treating for people who are depressed.
Thank you. I think now we are already going over time. So thank you so much, Dr. Penninx, for this fantastic talk and for answering all of our questions and even more. So the session will wrap up now, and I don't know, Dr. Williams, if you wanted to make some closing remarks.
Thinking we could, if you agree, Dr. Penninx, we could send you the questions we didn't get to. It's such a fascinating presentation. You've generated a lot of questions. But thank you.
Yeah, happy to take questions, and people can email me when they want to see certain papers or when they want to get better insights in certain things.
I really appreciate it. It's so impactful how you approach the heterogeneity, make it clinically meaningful, really run these incredible longitudinal studies with multiple measures, standardized assessments, and then the new trial, Enriching for Metabolic Depression. It's so fascinating. So thank you for sharing it with us.
You're welcome, and I wish you a good day.
Okay.
Thank you.
Good, and a good
time here.
You.
Thank you.
Thank you. Bye-bye.
Thank you. Goodbye.